My name's Darren, I work at the University of Exeter, and my title is DNA modifications in specific nuclei populations purified from human brain tissue. So most of my work is within the complex epigenetics group at the University of Exeter, and basically we're looking at epigenetic variation in the human brain and how it links into development and aging and various neurodegenerative diseases. So specifically in this talk I'll be looking at modifications of cytosine, so methylation, and also interestingly hydroxymethylation. So for example, it's been found in neurons that increased levels of hydroxymethylation have been found within gene bodies that correlate positively with gene expression. And we've also done another study looking at the distribution of hydroxymethylation across the human cortex and cerebellum. And so ONT is the perfect technology to look for modifications due to algorithms that can show the difference between bases and modified bases. So the human cortex is a very complex tissue. It consists of many different cell types, and traditionally we've actually looked at bulk tissue. The trouble with looking at bulk tissue is that all the different cell types have different different transcriptomes, for example, and you just get a kind of average. So these days we're moving on to single cell transcriptomics, so we can look at individual cell differences. So we've developed a protocol based on fluorescence activated nuclei sorting. So we take brain tissue from post-mortem brains, and we use cell-specific DNA markers to actually sort these into different cell types. And then we can use these different cell types for our various epigenetic and transcriptomic and genomic analyses. And then we can link these to various diseases. So this is our kind of laboratory pipeline. So in this talk, I'll be talking about two cell types: the neuron, which it uses a NUN marker, and the oligodendrocyte, which we separate based on a SOX10 marker. And so we purify DNA from our nuclei, and we sequence them on a promethine R10 flow cell. Basically, we usually put in 1 to 2 fan samples per flow cell. That gives us somewhere between 10 and 30 times mean coverage. And so in this study, we had 3 neuronal samples and 3 oligodendrocyte samples. And so this is our whole genome analysis pipeline. So we're using Dorado for base calling and looking at modification. We're using Modkit to extract the methylation data, and later in the talk I'll also talk about how we can phase the reads using Claire3 and WhatsApp. So looking at the profiles of either methylation or hydroxymethylation, we can quite easily separate out the sample. So this is just a simple PCA plot of all the 8 million shared CPG sites. So the neurons and the oligodendrocytes have quite distinct methylation, hydroxymethylation profiles. We can also look at them with heat maps. Again, that's another way of separating out the samples. Now, if we look at the distribution of methylation across the sample, so we've got percentage methylation and the density. So these are the oligodendrocytes. So we get 2 peaks, one around 0, one close to 100. So most sites are either unmethylated or completely methylated. If we compare this to the neuron, we still get a peak at 0, but we actually get more of a hump around 50%. And we can see the reason why this is if we actually look at the hydroxymethylation density plot. Again, the SOX10, the oligodendrocyte, has actually very little hydroxymethylation, so we just got a peak at 0, whereas the neuron has, again, a hump about 50%. So in the neuron, a large number of the CPGs are a mixture of sort of methylation and hydroxymethylation, whereas in the oligodendrocyte, they're nearly all methylated. So we created a reference set, so we just joined these 3 samples together, so we get sort of coverage of around 60x. We've got a UCSC track, which is publicly available. we've used this reference data set for the rest of the analyses, because what we wanted to do was to compare it to other technologies. So in our lab, we've used other technologies. Do we get similar sort of results? So I'm going to show you the difference between, or the comparison between ONT sequencing, where we get 28 million CPGs or so, give or take a few. Compare that against the Illumina array, where we've got around 850,000 CPGs. The twist, which is a kind of enriched whole genome bisulfite sequencing, we've got about 4 million CPGs. And also we've tried the KEGX hydroxymethylome capture sec, which doesn't give you sites, it just gives you enriched sequences. So again, we do some density plots here just to compare ONT with the EPIC and the twist. So this is for the EPIC. Again, we're getting sort of a bimodal distribution. We add the twist in. Again, bimodal. Notice the peaks are closer to 100 and 0, which is what we expect, the difference between the sequencing method and the array-based method. And then we add in the ONT, and it's very similar sort of distribution. Now, notice here, the twist and the EPIC cannot distinguish generally between methylation and hydroxymethylation. So to make it comparable for the ONT, I've added together the methylation and the hydroxymethylation, so we get what I call bisulfite. And notice when you do that, you don't see any difference between the NuN, which is the red line, and the SOX10, which is the dotted red line, because overall adding the methylation and the hydroxymethylation together is giving us the same sort of signal. We did correlation coefficient, so this is between ONT and EPIC, so around about 0.96 for shared CPGs, and the same, we compared ONT with twist, we're getting nearer to 0.99 correlation coefficients. Now, because the KEGX is a different technology, we had to compare it in a different way. So this just looks at enriched areas that are enriched in hydroxymethylated sequences, the pull-down sort of method, so you get peaks. So if you visualize this on the UCSC track, the top two tracks are from the KEGX, and you basically get just regions. So what I wanted to do was actually compare the average methylation from ONT within and without these regions. So if we do this, so in red we have the mean hydroxymethylation as measured by ONT within the KEGX peaks, and in blue it's the average methylation outside of those peaks, so they're significantly different, and you get significantly more methylation, or hydroxymethylation, sorry, in the neuron as compared to the oligodendrocyte. So this just shows the distribution of methylation, hydroxymethylation using ONT across the gene body. So the top line is methylation in the oligodendrocyte. The bottom line is hydroxymethylation in the oligodendrocyte, not much hydroxymethylation. In the middle, we've got the NuN sample, so we've got both methylation and hydroxymethylation, which are sort of higher across the gene body. So I finally wanted to talk about something else which is really great about ONT. Because it has long reads, we can phase them, so we can actually say whether the read comes from one allele or the other. And so this enables us to look at imprinting. So in imprinting, one allele is silenced because of methylation at an imprinting control region, and the other is active. So we use Claire-free to phase the reads, and Claire-free to look at variants, WhatsApp to phase the reads. So I'll just give you a couple of graphics here. So this is a known imprinted gene called Plagal1. So on the graph there, I guess the thing you want to look at is the bottom. You have 4 lines here. We have hydroxymethylation and methylation, and then we also have the 2 phases. It's probably simpler to look at the oligodendrocyte on the right-hand side, because there's not much hydroxymethylation, so that's the 2 lines at the bottom. The light blue area is the known imprinting control region, and you can see there one phase, the methylation has dropped greatly within the oligodendrocyte. If we then look, so we can see imprinting in both of those. It's quite interested in the neuron because the methylation has stayed high on the imprinted region, but the hydroxymethylation has dropped off. And this one's even more interesting. This is a cell difference in imprinting. So in the neuron, again, look at the highlighted blue region, that's the imprinted control region. There's no real evidence of any imprinting, whereas the oligodendrocyte on the right-hand side, you can see that dip in the red line on one of the alleles, the methylation has dropped off. And that fits in with other known work. The IGF-2R is imprinted in glial cells, but not in neurons. So just quickly go through my conclusions. So we've developed a FANS protocol to isolate the cell-specific nuclei. We're using ONT, we're getting nice high sequence depth. We can look at around 28 million CPG sites. The results we get are consistent with other technologies. We've been able to look at cell-specific differences in DNA modification. The levels of hydroxymethylation are higher in neuronal cells and oligodendrocytes. We've been able to also look at phasing to detect cell-specific imprinting. Future directions, we want to look at the link between methylation and hydroxymethylation and gene expression, because there's quite a lot of work showing that there is a link there, especially in hydroxymethylation. And we are going to extend the analysis to various other cell types based on our ability to separate them out. And I just want to thank members of the complex disease epigenomics group, especially Barry, who did the cell sorting, and Rosie, who did the sequencing. Thank you very much for your time.